First of all, I was reminded to remind you to use the hashtag MD22 if you are tweeting about this event. And then it's my great pleasure to welcome Mirela Delibegovic. Mirela is a professor at the University of Aberdeen. She's a chair in diabetic medicine and director of Aberdeen Cardiovascular and Diabetes Centre. Professor Delibegovic is also chair of the British Heart Foundation's Project Grant Committee, and she was recently elected member of the Royal Society of Edinburgh, which is Scotland's national academy. She did her PhD at the University of Dundee at the MRC Protein Phosphorylation Unit, and after a postdoc at Harvard Medical School, she obtained a faculty position at University of Aberdeen in 2007. Over the past 20 years, she has published many seminal papers, especially on the role of protein tyrosine phosphatase 1b, and this is also, I believe, the topic of today's talk. So the floor is yours. Thank you.
Thank you so much. And I'm so impressed that you were able to say Delibegovic several times. Thank you. Right. So thank you so much for the invitation today. It's absolutely wonderful to be here. And this is my first time in Copenhagen. It's a beautiful city. Oh, so thank you again. So I'm going to kind of start slowly, and I will be using a mixture of published and unpublished data, especially because some of the unpublished data is not in review yet, so I just thought perhaps I'll keep it back for informal discussions. So we all know that we are living in an aging society as well as obesogenic society. So I wanted to use some of the Scottish statistics here, but fear not, they're quite as bad everywhere else, although probably not here because everybody cycles. But in Scotland, about 30% of all adults are obese. And of course, 7 out of 10 of people are overweight and obese. And about 40% of people don't do any exercise or not much. So what my lab focuses on is different strategies to improve aging-induced outcomes, whether that's decreasing body weight, increasing physical activity, etc. We do this through pharmacological as well as nutritional interventions. I also wanted to use some of the British Heart Foundation statistics. So we know that heart disease is the major killer in the Western world. But actually, if we think about, and I'm going to work out if I can use this fancy pointer. Yes. So if we actually work out that other diseases that are associated with being overweight or obese, such as having type 2 diabetes or Alzheimer's disease and cancer, it probably accounts for half of all deaths, just some of the things associated with being overweight. So this is a very simplistic diagram of what I've been interested in the past 20 years or plus. And really it's kind of understanding why being overweight or obese leads to these disorders. So we know the white adipocytes don't just secrete leptin, but I'm using a leptin as an example here. From the perspective of PTP1B. So we know the leptin coming out of adipocytes is able to signal up to the hypothalamus to regulate appetite, to regulate food intake versus our energy expenditure. And we also know that on the other side of the story, the liver is a major source of glucose in our body. And when we eat and we keep eating more and more, the pancreas has to work incredibly hard to secrete more insulin to allow this glucose uptake into different peripheral insulin-sensitive tissues. So really what I've been interested in throughout my career is the state of when does insulin resistance develop and why does it develop and at which point can we interfere, and as well as the states of chronic inflammation within the body. So these are some of the things that we are interested in our laboratory. So in my very long-standing collaboration with Dr. Nimish Modi, who's at the University of Aberdeen, we were working on vitamin A-like drugs, so retinoids such as fenretinide, Foroxo, and similar. Recently, we've been more interested in dietary restriction mimetics with specific emphasis on essential amino acid restrictions such as methionine, and seeing some incredible effects in regards to methionine restriction. We've got projects working on synthetic cannabinoids in collaboration with AstraZeneca, looking at kidney health. That's one of the diabetic nephropathies, also one of the major killers associated with having diabetes. And more recently, we've been using patient-derived organoids from patients with HCC and NASH, and looking at different signaling pathways that we can interfere with in order to hopefully reverse the state. We also work on a number of drug discovery projects, and once that's patented, I can tell you all about it. But today, I will be telling you about the bread and butter of the lab, which is protein tyrosine phosphatase 1b. And the reason the PTP1B has always been so interesting is that it's one of the rare molecules that really is at the intersection between the leptin signaling and the insulin signaling pathway. And I wouldn't be from Dundee if I didn't at some point at the start of my talk show you a signaling cascade. So we know that leptin binds to the leptin receptor and results in phosphorylation of a number of targets such as JAK2, STAT3. And we know the insulin binds to the insulin receptor to stimulate phosphorylation and activation of downstream cascade to allow glucose uptake. So this is an example of one of the projects they've been working on, which is diabetic retinopathy and the role of PTP1B in that one. So what is PTP1B? So PTP1B was the first tyrosine phosphatase ever identified and purified by Nick Tonks in the 1980s. It is present in every single cell type in our body, including all the insulin-responsive tissues, and has been now implicated in almost every signaling pathway that one can think of. But from my point of view, it was always interesting because it was a validated in vivo insulin receptor phosphatase. So this is very, very old data, maybe older than some of the audience today. But this was the original phenotype of the global PTP1B knockout animals. So the animals were generated in late 90s, early 2000s, because PTP1B was thought to be a major oncogene. But what was a surprising finding was that these animals were actually resistant to diet-induced obesity. So when placed on high-fat, high-sugar diet, they did not gain weight, and their fat pads, as we can see here, are significantly smaller with no changes in brown adipose tissue. So this has kind of led to years and years of research and hunting by different pharmaceuticals, including Novo, trying to develop PTP1B inhibitors that would be cell permeable. So to kind of summarize years of our data, background data of kind of trying to home in where does PTP1B exert its effects and how does it do it? This is a summary of many years of work. So we were able, during my postdoc years, in collaboration with my colleague and a really good friend, Dr. Kendra Benz, we were able to recapitulate the global PTP1B knockout phenotype in the neuronal specific knockout mice. So we were able to show that if PTP1B was knocked out within all the neurons in the brain, that the animals could not gain weight, and that was actually on chow as well as high-fat diet. And what was even more interesting for us was that this was not only in knockouts, but also in the heterozygous mice. We were also able to recapitulate the massive improvements in insulin sensitivity in these animals, kind of highlighting that PTP1B is a major regulator of body weight and appetite, and that neuronal inhibition would be required to achieve its anti-obesity effects. But in regards to its anti-diabetic effects, again, this is years of work summarized in one slide. What we've been able to do is highlight that PTP1B regulates insulin receptor in vivo in the peripheral tissues such as the muscle and the liver. So muscle-specific PTP1B deletion resulted in no changes in body weight, but huge improvements in insulin sensitivity. And likewise, in the liver, we saw the same effect, but in the liver, it was more interesting because these animals were completely resistant to induction of endoplasmic reticulum stress as well. And from this work, we also worked out that PTP1B was a major regulator, not just of insulin sensitivity, but also lipid homeostasis and inflammation. And of course, neuronally, we knew that PTP1B was a regulator of JAK-STAT signaling within the hypothalamus. But in 2006, a paper from Michel Tremblay in Montreal came out to highlight that PTP1B had a really important role within the macrophages, and that actually inhibiting PTP1B may not be such a good idea. So in these experiments, they used PTP1B knockout fibroblasts, which then they stimulated with LPS. And we can see here as the knockout fibroblasts are stimulated with LPS, that you see an increase in production of nitric nitrate, an increase in phosphorylation of inducible nitric oxide synthase. And this was associated with hyperphosphorylation of STAT1. Furthermore, when they took wild-type and PTP1B knockout animals and injected them with LPS, there was quite a striking effect. So we can see here in the wild-type animals that were injected with PBS, there was no mortality. Wild-type animals that were injected with LPS had some mortality, but quite low, 20% over this period of time. However, animals, PTP1B knockout animals that were injected with LPS, some of them, well, 50% of them died within first 24 hours. So that was quite worrying. But that also kind of fitted in with some of the data that I had obtained when I was generating tissue-specific knockout animals, where I wasn't seeing just the beneficial effects of PTP1B deletion. So this was an example from the cross that I made to the AP2 CRE, which now we don't use. In the olden days, we used to think that that was a adipocyte-specific knockout. But as it turns out, these animals had really a phenotype that was associated with B-cell autoimmunity. So if you look at the example here of flow cytometry from wild type versus knockout mice, we can see that there's a huge increase in TER119 CD19 and CD5 CD19 positive cells within the knockout strain. And even more so, something that we observed in global PTP1B knockout mice that we only recently published was that these animals also had really enlarged spleens as well as the lymph nodes. So when I went back to check whether AP2 was actually deleting anywhere else, what I could see is that, yes, it was deleting in white adipocytes, but it was also deleting in bone marrow and peritoneal and splenic macrophages. So to cut the long story short, we had generated a adipocyte-specific knockout using adiponectin CRE, and that was nothing to write home about. It had absolutely no phenotype. And all the drug discovery efforts that were kind of targeting adipocyte PTP1B were pretty much failed projects, because actually adipocyte PTP1B knockout, if anything, leads to increased body weight and absolutely no changes in sensitivity. But for us, the inflammatory phenotype was something that was really interesting, and we went after this one. So we first generated myeloid-specific PTP1B deletion to really understand how PTP1B was regulating the LPS-induced mortality. So this is just to show you that we had a really nice deletion in bone marrow-derived macrophages, as well as in dendritic cells and all the myeloid cell compartments, and nowhere else. And the first experiment that we did was use these animals to inject them with LPS and see if they are indeed more susceptible to LPS-induced mortality, and also to check the effects on glucose homeostasis. But when we did the glucose homeostasis, the GTT test, we could see that there were absolutely no differences between the wild type and the knockout animals, just in response to LPS challenge. 24 hours later, however, we saw that half of the animals, because everything was done blinded, half of the animals were looking like they had signs of hypothermic stress, while the other half of the animals looked completely normal. So, of course, we thought that we were teasing out the phenotype that was observed in the global knockouts. But what was quite surprising is that the wild-type animals were actually still suffering from the LPS injection, while the knockout animals were completely resistant to it. And to cut the long story short, what we found is that these animals had suppressed secretion of pro-inflammatory cytokines such as TNF-alpha, interleukin-6, and MCP1. So the next challenge for us was, okay, so maybe LPS was not enough to induce this phenotype that we were expecting. So how about we put animals on high-fat diet? So we had animals on high-fat diet to induce chronic stress, inflammatory stress, and what we could see is that actually myeloid PTP1B knockout animals had significantly improved glucose homeostasis. So this was going against the hypothesis and previously published data from the global knockouts. So we did the same test as before, but now in the background on high-fat diet. So animals were fed high-fat diet, injected with LPS to induce transient insulin resistance. And what we could see is that in the wild-type animals injected with LPS, we can see increase in insulin levels, whilst this was completely suppressed in the knockout animals, and actually was suppressed to the level that was lower than the basal levels. So to understand the mechanisms behind this, we took out the bone marrow-derived macrophages, cultured them, stimulated them with a low dose of LPS, and looked at the time course of different genes that are induced at this time, using ultra-pure as well as standard LPS to kind of delineate between TLR2 and TLR4 pathway. But the data were the same in that the wild-type animals in blue had increase in TNF-alpha gene expression over time, and a decrease in IL-10 over time. However, in red, if we look at the knockout animals, we can see that TNF-alpha goes up, and then it's blocked at 4 hours. However, IL-10 secretion in these animals goes up and stays up even after 24 hours. Then we went back to our blood samples from the animals, and what we could see, we looked at IL-10 circulating levels. And what we could see is under basal conditions that lysine PT1B knockout animals were over-secreting IL-10. And when injected with that high-fat diet and the LPS dose, we can see that there's a super-secretion of IL-10. And actually, the IL-10 levels in these animals negatively correlated to insulin levels, as well as the ALT activity. So, with this knowledge, we were really interested in kind of inflammatory processes and diseases associated with inflammation. And like I said at the beginning of my talk, we were really interested in heart disease. Sorry. So we know that elevation in markers of inflammation predicts outcome in patients with coronary syndromes independently of myocardial damage. And we also know that treatments that reduce coronary risk also limit inflammation. So we wanted to know is if we could actually use the function of macrophage PTP1B to improve outcomes in atherosclerosis. And we asked this question in 2 different ways. We had 2 different experimental models of atherosclerosis. We first did a genetic cross by crossing our myeloid PTP1B knockout animals to the standard ApoE knockout mice that are susceptible to development of atherosclerosis. And secondly, for translational point, which everybody's interested in, we wanted to know what would happen with pharmacological inhibition of PTP1B using a more severe model, which is the LDL receptor knockout, which is kind of more close to human model. As Randy said, one has to use the mice to really understand what's going on. So, as I said, we crossed the animals, and first of all, we wanted to check that they had deletion, and indeed, if we look in ApoE background, we can see that we have deleted PTP1B in the myeloid compartment by about 80%. We had animals on chow, as well as high-fat, high-cholesterol diet to induce the plaque formation. Cholesterol has to be added in. So we used male and female mice. I'm sure you'll know that now that is the norm. We can no longer just use male mice because females are too complicated. So when we put animals on high-fat, high-cholesterol diet, we can see that whether they were ApoE or ApoE lysine PTP1B, there were no differences in body weight whatsoever. If anything, in females, they're slightly heavier, and males look slightly lower, but the numbers were high enough to know that there were no statistical significant differences. Likewise, using echo-MRI scanning, we could see that there are no differences in fat mass within triple knockout as opposed to ApoE. So what was quite nice for us was to see that there were big improvements in glucose homeostasis. So these are the data on animals on high-fat diet for 12 weeks. We saw these differences much earlier on, I should say, as well. We can see that both in males and females, we have beautifully improved glucose homeostasis. So just deleting PTP1B from the myeloid compartment on this ApoE background is enough to completely rescue the glucose homeostasis phenotype. And it was really nice to see this in the females because, if anything, in the females, they weighed a little bit more and had a bit more fat mass, but this wasn't statistically significant. So we could say with confidence that these improvements were despite absences and differences in adiposity. Furthermore, we could see that there were lower insulin levels in both males and females. We could also see that serum cholesterol levels in males and females were significantly lower. Now, this was interesting because the only time we saw differences in lipid levels in regards to PTP1B and its function was when we had generated the liver-specific knockout mice, as I said earlier, that were resistant to endoplasmic reticulum stress induction as well. But we also saw the serum triglycerides were significantly lower in the triple knockout animals. Going back to the IL-10, so we know that IL-10 is expected to improve insulin sensitivity, and we wanted to know whether this was the reason why the ApoE lysine PTP1B knockout mice had improved glucose homeostasis. But actually, there were no differences, significant differences in IL-10 between the ApoEs and the triple knockouts. But using our global lipidomics analysis, as well as targeted analysis looking at prostacyclins, what we found is that the levels of vasodilatory PGE2 levels were significantly increased in these animals. And that was again interesting because we had a whole load of unpublished data from previous years seeing that PTP1B in the liver was causing differences in PGET and COX-2 induction. So this was kind of driving us towards the new phenotype that we had not anticipated before. So finally, as I said, the reason we started this experiment was because we wanted to know whether PTP1B genetic deletion in the myeloid compartment would rescue the phenotype. So we went to assess the atherosclerotic plaques within these animals. And we can see here that both in the males and in the females, the plaque area is significantly improved. What's really interesting to see is in the females, this is really much higher, but we can see that the triple knockout is completely protected. So then we went to the translational experiments, asking, okay, so it's all genetics, so what does it matter, really? Nobody's born without PTP1B in myeloid compartment. So we went looking for the best inhibitor that we can find, and we found Genera Corporation inhibitor MSI-1436, or trodescamine, as it's also known. It was purified in the 1980s from shark livers, and it was basically published to have really, really beautiful effects on body weight. So 1436 was compared to food restriction, and what you can see here is that PTP1B inhibition is equivalent to caloric restriction in regards to reduction of body weight as well as body fat. And if anything, it's actually better at reducing body fat than caloric restriction. And it's associated with decreased food intake and increased VO2. So we decided to use this one, and we also needed to know what dose to use. So this is just one single poster that was published in humans using this inhibitor, which is the pharmacokinetic study of trodescamine, or MSI-1436. So we can see here in humans, one single IV infusion or different doses. So this is 10 milligrams and 5 milligrams, 10 and 20 milligrams per kilogram. We can see that there is really kind of nice pharmacokinetics, and it takes about 36 hours to clear it. But the nice thing here is that also people who were on 10 milligrams per kilogram dose of trodescamine or MSI-1436 had significantly lower glucose levels after a single dose of trodescamine, as well as insulin levels. So we decided to go with this one. So we got our 90 LDL receptor knockout animals and split them up on chow versus high-fat, high-cholesterol diet. And what we did is a two-way approach. We placed all the animals on the diet for 2 weeks and then gave one set of animals PTP1B inhibitor, once a week IP injection as our chronic treatment. And then a second question was whether we can actually reverse the pre-established plaque development. So in this case, we fed animals for 8 weeks of high-fat, high-cholesterol diet and then started the treatment at this point in time. And we knew that 8 weeks was the appropriate time because we'd done pilot experiments to see how long it takes to develop beautiful plaques within the aortas. We did a number of echo MRI scans, glucose homeostasis assessment throughout the study. So I'm showing you data from both chow and high-fat diet, and I'll concentrate here on high-fat diet data. So we can see here that the LDL receptor knockout animals that are just injected with saline keep gaining weight on high-fat, high-cholesterol diet, as expected. And here at week 2, in blue, these are the animals that start getting once-weekly injections at actually 5 milligrams per kilogram dose. So what we can see, as soon as they start receiving the PTPI inhibitor, their body weight is suppressed. And actually, at this point in time, in standard with UK Home Office regulations, we had to stop treating our animals, okay, because the animals were losing weight and the animal facility wasn't happy that this wasn't going to continue. So we actually stopped, but we kept the animals. So these are now subsequent 6 weeks. So 6 weeks on with no treatment, the animals remained at that suppressed body weight. Now, for me, the most interesting treatment group here is in red, which is our acute one single dose, just like it had been given to the humans. So we gave one single dose after 8 weeks of high-fat, high-cholesterol diet. We could see almost immediate suppression of body weight, and we no longer injected for 4 weeks. And even 4 weeks later, that suppression of body weight was completely there. We can see here in chow animals that even on chow animals, when you give them PTPI inhibitor, they basically just do not gain weight. That's it. They just drop and remain at that point. So doing the echo MRI scanning, as I said, throughout the whole experiment, what we could see is that in the animals on placebo, they keep gaining fat. Animals that are on chronic treatment have decreased body fat. And then, as I said, this is the washout period where they're receiving nothing, but still the fat is completely gone. And in the acute dose treatment, we can see that the fat mass goes down and it remains down after a period of no treatment. And likewise, this is just controls on chow. We saw, as before, it had been published in the OB mice, that this compounds suppresses food intake. So we can see here in the chronic treatment animals, the food intake is significantly lower, and in the acute treatment, even more so. Looking at glucose homeostasis assessment, I mean, one would say you've got decrease in body weight, so of course you're going to be seeing an improvement in glucose homeostasis. We can see here that in blue, animals on the chronic treatment have significantly improved glucose homeostasis at week 8. But what's really nice is in this red line, when we have the acute treated mice, we have signs of improvements of glucose homeostasis, which are significant, but they remain so, are even more striking 4 weeks later, after they've not been receiving the treatment whatsoever. Now, this may not be very attractive for pharmaceutical companies because nobody wants to just give one single shot of something and then that's you sorted forever. But when we look at other circulating markers, we could see that serum insulin levels, cholesterol again was significantly lower, and triglycerides even more so. So I have to say we weren't that surprised when we sectioned our aortas to find that there was a beautiful decrease in the plaque area in both chronically, but really importantly in the acutely treated mice. So going back to my signaling and kind of just expecting what we would be seeing, we went to the, we had a massive cohort of mice because some were being sectioned, some were being used for signaling. So we were just expecting to see improvements in insulin receptor phosphorylation as we'd seen before, kind of with muscle, etc. But what was quite surprising, so these are animals, as I say, that were injected with insulin at the terminal procedure, but these ones had 6 weeks washout of PTPI inhibitor treatment, and these one had 4-week washout. And we didn't see much difference in insulin phosphorylation. It was pretty much the same. What was quite interesting, however, was that AKT was hyperphosphorylated, especially in the acute treated mice. And then there were no differences in other markers. But importantly, there are no differences in PTP1B levels because MSI-1436 or trodescamine inhibits PTP1B activity, but does not affect the levels of PTP1B itself. But what was surprising, and the only time we found the PTP1B was regulating phosphorylation of AMP kinase was in the neuronal knockout mice within the hypothalamus. So this was just control blots that were done, but we actually saw huge phosphorylation of AMP kinase at the threonine 172 site. And this is obviously really significant here in the acutely treated mice, probably because the washout period for these animals in chronic treatment was 6 weeks, so it's a bit longer. So with this knowledge in mice, the reason I was interested in this was because of this paper, which showed that in human patients with end-stage dilated cardiomyopathy, this was a paper from Circulation, they found decreases in JAK and STAT3 signaling in DCM samples. So if you look at here, phosphorylation of JAK2 in patients with diabetic cardiomyopathy, it's completely gone. So phosphorylation is not there whatsoever. And importantly, TYK2 as well. And TYK2 is known to be also a direct substrate of PTP1B. So if you look at phosphorylation of TYK2 in DCM patients, it's gone. And likewise, while there are no differences in STAT1, differences in STAT3 in these patients are huge. So we wanted to know whether we can translate our knowledge from our mouse models into the human models. So the first experiments we did was just recruiting volunteers. And at this point, these were just healthy volunteers. So and this is the work from Oliver Health, who was a clinical fellow doing a PhD in my lab. So he collected monocyte-derived macrophages from human volunteers, and just a really simple p-tire Western blot. Okay, he stimulated cells with MSI-1436, and we can see that this resulted in hyperphosphorylation of many targets. So when he looked at whether the targets that we are interested in, which is TYK2, JAK, STAT, and AMP kinase, we can see the MSI-1436 or trudescamine directly hyperphosphorylates TYK2. And I can see my chair is up, so I'm going to speed up. Okay. And what he also found is that this was associated with hyperphosphorylation of STAT3, phospho-AKT, importantly, and importantly, phospho-AMP kinase. So the PTP1B inhibitor by itself phosphorylates AMP kinase on this site. I'm almost done. So we recruited 60 volunteers, 30 who are healthy volunteers and 30 with severe atherosclerosis from GP clinics around Aberdeen. And what we found, as we expected, he cultured monocytes as well as monocyte-derived macrophages from these patients, and we didn't see any differences between the wild type and the healthy and patients with atherosclerosis in regards to PTP1B levels, and no differences between males and females. However, what was really important was when we use oxidized LDL as our stimulus of atherosclerosis. So here he looked at JAK2 phosphorylation. So if you look at healthy volunteers and their monocytes, when oxidized LDL is used in these, we can see increase in JAK2 phosphorylation, but it's not significant. But if oxidized LDL is incubated with trudescamine or MSI-1436, this is hyperphosphorylated. But this becomes even more significantly so in patients with atherosclerosis. And likewise, when we look at STAT3, the differences that we're observing are not huge. We do see improvements in phosphorylation or increase in phosphorylation with PTP1B inhibitor treatment in healthy volunteers, but this is significantly more evident in patients with atherosclerosis. So with this, I'm going to finish, and hopefully I've been able to convince you that PTP1B is still a really, really good target for treatment of obesity and type 2 diabetes, but also cardiovascular disease. But one has to be quite careful where they're targeting it. So thank you so much, and I'd just like to thank my lab, who did all the work.
Thank you very much. Very fascinating talk. I think we have a question on YouTube.
Hi, Mirella. Yeah, so over here, down on the right. Yeah, ground floor. Yeah.
Oh.
Hi. So we have one. We've got a question from YouTube, and apologies for the name. Ramasima asks, have there ever been any studies done on calorie restriction and downregulation of PTP1B?
Yes, calorie restriction does decrease PTP1B levels, well, at least in our hands, and also PTP1B activity. And so do a number of other dietary interventions, actually. Yeah.
I'm Kei Sakamoto, CBA. Wonderful talk, Mira. Thank you very much. So, a couple of questions. So, I was quite fascinated that PT1B1 appeared to kind of control the IL-10 secretion, and so that also you showed the myeloid-specific knockout that you had a boost. So that's so is it Toll-like receptor 4 dependent pathway, and then do you know the target that control pro and anti-inflammatory cytokine secretion?
Thank you. That's a good question. So we think that the secretion of IL-10 is driven by hyperphosphorylation of STAT3. So it had been shown before that STAT3 hyperphosphorylation leads to induction of IL-10, which actually, when we think about pro-oncogenic signaling, is not a good thing. And we have done experiments where we use RUX, so the STAT3 inhibitor, and when we use the STAT3 inhibitor, that hypersecretion of IL-10 is completely ablated. And also the phenotype that is associated with how the cells move and how they behave. So we think it's STAT3 driven.
Just one more question. So you are inhibitor studies that, and so in terms of the body weight phenotype you have, of course, an obvious question is whether this is, kind of a neuronal effect on food intake, because you have acute effect and long-term effect. And then also you see also, increases in IL-10, for example, circulation under this context.
Yeah. So again, another good question. And we've done studies since then where we actually also looked at dose. And we've also used some unpublished PT1B inhibitors to compare the effects in ApoE background. So what we find is that the dose we use, which is the dose which is half of the human, is probably too high, because even at 1 milligram per kilogram, we see huge improvements in the phenotype of, in regards to atherosclerosis, but we see no differences in body weight. So we've started now delineating the effects of body weight from the effects on metabolic health. And what was the second question? Sorry.
So if you treat with a drug, do you see also the boosting of the IL-10?
So yeah, we don't. I did show those data. It looks like it's slightly upregulated, but considering we had 90 animals, it's not significant. So IL-10 is not induced by this drug treatment. And I should also say the other inhibitors, unpublished PT1B inhibitors we have used, are also showing really promising effects in regards to atherosclerosis. So PT1B is certainly driving that. And we do actually have hopefully a novel mechanism that we think is behind it, but that's something I can tell you maybe in private. There's a couple of questions, so Maureen.
Thanks, Bernard. Great talk. I was intrigued by your statement that when you do the fat cell-specific PTB1 knockout correctly, you don't see any phenotype because, as you well know, if you modulate insulin signaling through other mechanisms in fat, you see both autonomous as well as non-autonomous effects on metabolism. We
were hugely disappointed. There was a work of a PhD student for 4 years, and he did every experiment under the sun. The mice weighed slightly more, and you would expect if you got improvements in glucose uptake, then maybe you would have increase in body fat, and that had been shown with some antisense oligonucleotide work that was targeted to fat. But we didn't see any improvements in glucose homeostasis, no effects on food intake. We cultured isolated cells, did lipolysis assays. Nothing was changed in regards to wild type.
So you actually didn't see the biochemical changes in fat cells, or you saw those changes and they didn't correspond to in vivo changes?
Yeah, basically, everything was as it was in the wild type cells. So isolated primary adipocyte work, everything was exactly like in wild type.
So is PT1B not a regulator of insulin signaling in fat cells?
We believe that it's not.
Thanks.
And certainly not insulin receptor phosphatase in those fat cells.
We have time for one last question.
Great talk, Mirella. I was just interested, when you do the pharmacological inhibition, do you see any ill effects there? You might expect some impaired immune function. I'm just thinking of the translational aspects eventually taken into humans.
No, good question. We didn't see any any bad side effects. If anything, once the mouse was opened up, it looked like all the fat had been sucked out of it. Okay, so we saw improved liver health, everything that was positive. We didn't see any immunological phenotype that we should be worried about. I mean, you know, we can talk about genetic knockout and some of the ill phenotypes that we did see. So, for example, lice and PTPMB knockout mice, after they're aged, over 12 months of age, they actually start developing acute myeloid leukemia. Interestingly, only if you have 100% knockout. If you have 80% deletion, so 20% remaining PTPMB activity within myeloid cells, animals have improved metabolic health without any ill effects. So it's really an on and off switch. And that's why it's really important to regulate PT1B appropriately. So the nice thing about PT1B inhibitors is that they're never going to inhibit 100% of 100%.
And any effect on mTOR signaling? Sorry.
Oh, yes. Oh, mTOR. Yes, we do see really nice effects in regards to mTOR signaling as well as growth hormone-induced mTOR signaling in regards to PT1B. Yes.
All right. Thank you very much.
Thank you so much.
Very nice.